So genetic switches are able to control how much a specific gene is expressed at any given time. And when you consider some of these master body building genes, like those that we talked about earlier with the Hox genes, then you see that you can have numerous switches that can turn genes on or off depending on the tissue they're in, the stage of development they're in, etc. So here you have three examples of three different switches that are turned on at three different times. The E is when it's an embryo, the L is when it's to develop the larval legs, and the W is to develop wings in flies and butterflies. But in butterflies, this switch is turned on one more time. There's another transcription factor or some other protein that binds to this switch that brings in the RNA polymerase that turns on the gene one more time and allows it to develop these eye spots. So how much the gene is expressed, by changing the switch, you can change how much that particular gene is expressed over time. So the more you have, the more you express it, the more you change the number of eye spots or the size of the eye spot on the butterfly wings. So I'd like to take a moment and discuss the Hox genes again, because I don't think I did it justice. The Hox genes are incredibly important, and just to remind you, when you're looking at the development of an embryo, and this is an embryo of Drosophila melanogaster, which is a fruit fly, and this is the embryo of a mus musculus, which is a mouse, you'll see that there's Hox genes in both. There are more Hox genes in the mouse embryo than there are in the Drosophila. But these groupings, where they're located, where the genes are located, are incredibly important to what is going to be expressed when and where. So in vertebrates, it turns out that their position, there's a number of notes you should think about. That where the gene is, which one is turned on and which one's turned off, it's turned on and off depending on when the embryo is developing, where the gene is located. For instance, the posterior ones have priority over the anterior ones as far as turning them on or off. And then that also starts to develop certain characteristics. And if you turn certain Hox genes off, you can actually get homologies to other organisms. So you get all kinds of interesting control mechanisms from these Hox genes by looking at several different ways of handling turning these genes on or off. As you get DNA, a good part of these Hox clusters are controlled by chromatin activation or inactivation. Remember, the chromatin is usually how DNA is stored or is unwrapped, and if you methylate it or keep it wrapped up around the histones, then what you're going to start to get is you're going to get inactivation. In other words, you can't access. Remember, you have to unwrap it in order to access it and make mRNA. The yellow little lines here are mRNA. So in order for it to be active, it has to come out and be unwrapped from its histones. And so what you get is this unwrapping, and as the DNA is unwrapped, parts of the DNA that don't code for a protein act as cis elements. In other words, they act as inducers of mRNA production. They help the promoters bind the RNA polymerase to start making mRNA. And what you get is mRNA production if they're unwrapped, if they're if the chromatin allows for the activation of the DNA, the access of the enzyme RNA polymerase to the DNA. And so what you get is you have to expose both the cis element, the part that's going to help the gene make the mRNA, and the gene itself. And we've talked about translation forever, so you should have a pretty good idea what we're talking about here. So uncovering the chromatin, letting it unwrap or not, is one way of controlling the Hox genes. And in fact, every gene. In fact, this is a really good model for looking at control mechanisms for every gene. When we look at the different kinds of control mechanisms, another thing that we had talked about earlier is alternative splicing. Alternative splicing can, in this case, in at least one example of the Hox genes, can lead up to 6 different DNA products, right? 6 different gene products. So one gene can give you 6 different proteins, and we discussed this possibility earlier in the year. So depending on how you splice that message that was first transcribed from the DNA, you can get up to 6 different molecules that then go ahead and have 6 different impacts on the development of the organism. So this should start giving you some idea of how you can get from one cell to 1 trillion cells, how you can go from one cell that can make anything to a very specific cell like an eye cell or a brain cell or a lung cell or a liver cell. That DNA is controlled by all these different processes, unwrapping the DNA or not, methylating it or not. You can, you have alternative splicing. So if you make one gene, maybe that gene promotes the production of a protein. Maybe that protein that it made another gene produce another mRNA product. When you alternately splice the same gene, you get some other protein that maybe doesn't do as well in making that protein product, or makes a completely different product that slows down the production of arms or legs or changes it in some way. So these different tissues, the same gene can act in different ways depending on how the mRNA is spliced, how it's put together, how the exons are removed, how the introns are put together, and how the introns are removed. You can get different protein products, different gene products, depending on which promoter region you use. So if you use one promoter region, you get one protein product. You use another promoter region, you get another protein product. So you can regulate which promoter you use, and that will give you a different protein product. And so perhaps it's able to make the protein better, and so you produce more of it. And if you have more of that hormone or more of that growth factor or more of whatever it is, you have longer limbs or shorter limbs or more vertebrate or less vertebra. You can alternatively add poly-A tail, a length of poly-A tail. Maybe you add the poly-A tail here versus adding it here. So 2 different places you can add a poly-A tail, and if you do that, well, it changes again. It makes that protein, that product of that gene, different. And finally, you can, or actually it's not finally, there's a couple other things you can do. You can add and start to make in certain tissues or certain times during development, you can produce long non-coding long non-coding RNAs that bind to the mRNA and do not allow it to translate that mRNA into a protein. So you can literally turn off the gene that way as they bind to the transcription factors, these long non-coding RNAs bind to the transcription factors that slow down or stop the production of mRNA, and therefore you don't have any protein at all. So you can turn these genes on or off, these master genes on or off that way as well, which then changes what it is that cell is making, what kind of cell, what kind of structures, what kind of traits that particular tissue is gonna create. So you get this cell, these group of cells, they all started from the same cell, they all have the same DNA, but depending on what signals it gets from the environment, these different chromatins unravel or they don't unravel. They're expressed because you can access the gene or you can't use one promoter or another promoter. You add a certain poly A tail or you add it to another part of the mRNA so that you have a different mRNA. You can splice the mRNA differently. You can add a long non-coding RNA that's going to bind to the transcription factors that slows down or stops the transcription of the mRNA. Or you can even create these things called microRNAs that bind to the mRNA and degrade it, break down the mRNA, or bind to the mRNA and don't allow it to bind to the ribosome, so you never make a protein. And finally, there's been some examples where there have been some changes to the proteins and that combine with RNA to make ribosomes that actually impact the how that ribosome is able to translate parts of these Hox genes, so that it's just one more level of control where you can get specific structures to be made, one more level, one more opportunity for evolution to come in and change a structure, change a being, change what something ends up looking like in the end, and therefore may give it an advantage or a disadvantage depending on what it is. So, nuclear dynamics, again, the chromatin unraveling or non-unraveling, chromatin remodeling, changing which part of it is in the active region or is in active stage versus non-active stage. Transcriptional regulation, so you have alternative splicing. You have again that long non-coding RNA that can bind here it is, long non-coding RNA regulation. You have RNA processing. How do you splice that RNA? You have microRNA, which is really small RNA. They can either degrade the mRNA or stop the ribosome from binding to it and making that protein. And translational modification, so where you see these changes that change how the ribosome interacts with the mRNA. So all these things are ways in which we can control. And don't forget that there's a time, that the interesting thing about these Hox genes is that there's time. They're time sensitive, so depending on what stage of development the creature is in, certain genes are turned on and others are turned off. There's position. So depending on where it is, these might be turned on because they're posterior. These might be turned off because they're anterior, vice versa. There are all these other factors that have to do with where the cell is located and what kind of chemical signals is it getting from its neighbors and from its environment. This can all impact how they differentiate, how you get a single cell turning into some other cell, some other tissue. So development of creatures is impacted from DNA and how it's inherited in this way and is controlled in this way. And there's all kinds of places where that control can change the structures that they form. Another key concept is this idea of these switches that exist upstream from the gene that turn the genes on or off. So what we've been talking about is evolution and development. How evolution impacts development and how development can be controlled through various steps in evolution. And what kind of evidence is there in development for evolution? So all that, to be short, is Evo, Devo. So we've talked about for a while now that how Hox genes impact animal development. We know that there's this modular component where certain, the thorax, the abdomen have different groupings of these Hox genes. And because of their position, because of when they start to develop, because of timing, because of chemical concentrations of different transcription factors, because of all of these things that we just discussed and have been discussed in the last two videos, you get these Hox genes specifying what structures are made when, and so they really help control the development of the organism. They really are the regulators of the development. They're the ones turning things on or off and being themselves controlled by other factors. So what we've looked at is how you have these Hox genes in both an arthropod, something without vertebra, something that has a small brain but has an exoskeleton, doesn't have a closed circulatory system, very different evolutionarily, very far removed from a vertebrate like a mouse, a mammal like a mouse or a human, and yet we have the same exact genes, just a mouse has more of them and is controlled a little bit slightly differently. So because they have more of them and they're controlled slightly differently, you can make different structures, and they can be more complicated, like the vascular system, like the respiratory system. So what you get is control, the same DNA, having control over many different tissues. So these mechanisms of control are ways in which you could explain how the same DNA could, by adding layers of control, by adding layers of tissues, you can go from a simple creature like something like a fly, like an insect, like a single-celled organism, and keep building layers of control, layers of complexity until you get something like a mouse. It's fairly strong evidence, and you would predict something like this if something like evolution were happening. I mean, I don't know if anybody could have guessed that this is what was happening, but certainly it seems logical that if you're going to have this, if you find in this simple form this kind of control, and these two shared a common ancestor, that they would keep that control, that these conserved regions, these Hox genes are conserved, and meaning that they have almost exactly the same DNA, it's just controlled differently, and they're arranged differently. So that change in arrangement and control mechanisms led to a difference in structure. Also, the idea that you can get small changes, like a change in a switch or a change in orientation of the Hox genes, can lead to different patterns, different structures that can then lead to variation, not just genetic variation, but physical variation. And if there's differences in variation, some things are taller, some are shorter, some are slower, some are faster, some are red, some are yellow. If those are true, if those exist, those differences exist, then one could select one pattern over another. If it's not us, it could be a raven that decides to eat this one over this one, because this one, they think this is a predator looking back at them, and they think this is pretty, so they eat this one. So there could be, as long as there's a difference between them, then natural selection can select, doesn't necessarily have to, but can select one over the other. And therefore increasing the number of alleles of the one that has a benefit, that is best fit to survive, and decrease the alleles of the other, because if this one gets eaten and this one doesn't, then the next generation you'd expect to see more of these traits because this one's genes are going on with all its Hox genes in their proper place and all the molecular switches, gene switches where they're supposed to be. Then this is the one that you're going to see more of, and you're going to see less of these in the next generation. So again, when we talk about genes, remember we're talking about strips, little stages of DNA that through transcription and then once it leaves the nucleus through translation, produces a protein, some kind of amino acid polymer. So where would you find the control? How would you control that if you had to increase, if you make more of this protein, maybe if you make more of this protein, it's a growth hormone and the thing is taller. Maybe that protein is a dye, is a pigment. So if you make more of that protein, the redder the flower is. The more eye spots you get. The less of that protein, same protein, there's no mutation in the gene itself, so you can have the same gene. But you can make less of it. Well, how would you make less of it? Well, you could remember there's switches. There are switches upstream, switch 1, 2, and 3. You can turn those switches on or off. You could, remember there are these things called transcription factors. Transcription factors that bind to the switches, that bind to the promoter region. There's a promoter region. Bind to the promoter region. They stop what? They stop or help RNA polymerase a complex of complex and enzyme complex to bind to the RNA at the promoter region. So these transcription factors, these switches help that protein make what it has to make. You have spliceosomes that are going to cut out introns and splice together exons, and you have microRNAs that bind to and degrade mRNAs. Of course, you make the mRNA, can make several different kinds. You have ribosomes that make the protein from the mRNA. So at each one of these stages, at the transcription factor stage, at the gene switch phase, at the RNA polymerase phase, at the promoter phase, you have alternate promoters, for instance, that could change that. Spliceosomes, where you get different gene products depending on how using alternative splicing. Oh, let's not forget the poly A tail. You're adding a poly A tail. And depending on where you put that poly A tail, the position of that poly A tail could change depending on what you have. Again, all these are control points, are places where we can't control, that evolution can't control, can change what this protein looks like at the end, and therefore what it does and what it makes, because proteins are the things that make things, right? So you can use microRNA to stop the mRNA from making a protein. You can control it at translation. You can change which protein goes into the ribosome, or stop a protein from going into by down regulating that particular protein in the system, so that this protein doesn't match up with that mRNA as well as it used to and doesn't make as much protein. All of these things can change, all eight, can change or stop a protein from being made. Oh, and don't forget, there's another big control. That's just from transcription to translation. Remember, you have the chromatin control. It's with a Roman numeral 1. Whether that chromatin, that DNA, is unwrapped and allows the RNA polymerase to enter or not, or to match up with the gene or not. So you have chromatin control, you have transcription factors, you have at RNA polymerase, you have here at the gene switches, which are upstream from the gene. You have the promoter region, which is upstream from the gene. You have different spliceosomes. Of course, you have mutations that can change the protein itself. You have transcriptional control, so you have things that you can add to polytail or take it away. You can turn on or transcription on and off by having the transcription factors or not. You have microRNA, which could then destroy the mRNA product, the RNA product or not. You have translational control, so you can down-regulate translations to slow it down or speed it up. So it changes the protein, and by changing or stopping the protein, by changing or stopping the production of that protein, you're changing the development of that organism. You're changing it from black to white, red to yellow, big to small, or vice versa. Just by changing a single protein. And imagine that this happens with every gene. And remembering that this gene, depending on how you splice it, can make 6 different proteins. You make one protein, it does, and in each tissue, in each cell, in each region, these proteins can do different things. So, and if you have to go back to our discussion on hormones, hormones are made by the body. There are, things like insulin, glucagon. There are hormones that will do one thing in one tissue and something else in another tissue. That's why taking testosterone, for instance, is not a good idea to build muscle. Will testosterone as a hormone encourage the production of muscle, encourage this process to happen in muscle fibers and build you muscle? For sure it will, yes. Those anabolic steroids will build muscle. That's guaranteed. That's true. Depending on how much you eat and how much you work out, you'll build more and more muscle. However, those same anabolic steroids will do all kinds of other things in your body, because every cell type, every tissue is going to be impacted by all these processes differently. And in some, it might cause you to have cancer, higher risk of cancer. It could, if you're a male taking anabolic steroids, it could shrink your testicles. Stopping the extra anabolic steroids in your blood can start the growth of breast tissue in a male. These are all things that can happen because of this complex web of interactions of all these different molecules and all these different steps that we've looked at, studied, and have been tested on. So when we think about how gene expression can change morphology, how can gene expression change what something looks like, I think at this point it should be pretty obvious that development depends on these proteins. These proteins do all the work. So if you change the protein, you change the organism.